Thank you so much, Frank. And thank you to the whole team of the SPF for the great work you're doing and pushing forward to, beat this disease and raising funds and all of that. It's going to be hard for me to follow these really excellent talks we just heard. Very informative, and I could not agree more with the things that were said. What I will do over the next 30, 40 minutes is talk specifically more about genetics of HSP. I don't want to get lost in going through the many novel genes that are out there. This gets very technical very quickly, but hopefully give an overview over the sort of topics that move us and the general progress and the general challenges we face. And as a reminder, HSP, as we understand it today, is really a genetic disorder. So we have to, in the end of the day, we have to understand the genetics that causes HSP. Okay, so to sort of start with a 101 biology, because the term DNA or gene or genetics, it can quickly sort of sound very technical and very abstract. Here's a sort of simple drawing of how at the core of our bodies how we work. Let me 1 second here. So I hope you can see my cursor. So I can point things out. This round structure here is what we call a cell. And each cell, our bodies are made out of individual cells that work together. And a type of cell is, for instance, a nerve cell or an upper motor neuron cell. And you heard about these kinds of cells a lot just now. And each cell really contains a structure called the nucleus. It's sort of a special compartment surrounded by a membranous border, and so it's a sort of very protected compartment inside of each cell, and it is protected because it contains the most important piece, arguably, which is the DNA. So DNA is essentially a molecule, a very large and organized molecule. And it's so large, in fact, that it has to be packaged in a very specific way. And the way of how DNA is packaged in the nucleus is called chromosomes. So we have several chromosomes that when you sort of unpack them, you see that you sort of can unroll them, and eventually you would end up indeed with a single sort of long strand, and so at a very high magnification, you see how this strand really consists of a backbone, called as a backbone here on both sides, and then these 4 base pairs: guanine, cytosine, adenine, and thymine. Okay, again, very technical, but the point here is that these four molecules here, the order of which they appear in this strand is we realized 50 years, 60 years ago, is actually a code, is a sort of a secret code of biology of all life. And so understanding this code is really what will help us understanding how HSP works in the end of the day. So to confuse you some more, sorry, we actually consist out of not just a few cells or a few thousand cells. There are 40 trillion cells. This is an unbelievably big number. 40 trillion cells typically make up a human body. And about 100 billion of these are nerve cells. And a fraction of these 100 billion are actually motor neuron cells. So, as I said, each of these cells has a complete copy tucked away in its nucleus, the entire genome. Okay, so this long strand is in every single cell in the nucleus. And this strand, it consists of 3 billion letters, 3 billion of these ACT or G molecules. So, and what is also quite fascinating is that all life on Earth, as we know it, uses the same secret code of biology. So here's this illustration again. Now, what sounds like an impossible problem to work out, like reading the 3 billion letters in a single person, is now actually a routine in many molecular labs. And this is only possible because of developments, technical developments in the last 1 or 2 decades, really. And when you do that, when you actually take from a single person, average person, and you read what is in these 3 billion bases or letters, you find that at roughly 4 million positions, an individual has a variant that is different to what most other people have at this position. Okay, so out of 3 billion, about 4 million positions in your genome are sort of unique to you. That's important. And because it's important, because when we, for instance, study a patient with HSP, we study the genes. In theory, we have to consider 4 million positions in each individual, and have to decide, could one of these 4 million actually cause HSP? Now, these 4 million are spread across the entire genome, and in the genome, sort of in irregular spacing, we find about 20,000 genes. These are sort of areas of the genome that are especially meaningful. These genes, they make up, in the end, the proteins that construct our bodies. So these are the most important piece, but they only make up about 1 to 2% of the genome. What is also fascinating is that 99%, so when we look at many people in a population, let's say we look at 1,000 people or 1,000,000 people, we find that 99% of all the variation we see, 99% of all these 4 million variants are actually really rare variants. We see them only once or twice or 3 times in the patients we are studying, which makes it a little bit harder for us to understand their meaning, because it's not the same variants that occur again and again. It tends to be new variants all the time. Okay, and also an interesting side fact is that the majority of variants in human genomes are actually quite specific to your ancestry, where your family has originated from Europe, from Africa, from Asia, a very specific variants there. So cost matters also when studying HSP, when doing research. And luckily for all of us, the cost for sequencing or analyzing a human genome has really come down dramatically. It would have cost 20 years ago about $100 million to study a single HSP patient, the entire genome. Today, we can do this for about $1,000. And not at least because of this cost reduction, many studies in HSP and many other diseases have been performed. And here you see a map that the NIH gives out every year with the genetic associations and disease. And you see the immense progress. 20 years ago, this map would be largely empty. So how many HSP genes are there actually? As you know, there are many. And here is a map that is actually also not quite up to date because it's about 6 months old. There are now new genes in addition to that. And I can assure you that even for somebody who works in the HSP gene and does genetics in HSP, it is even for me hard to remember all the genes that are there. So how many are there? That's the big question. And really nobody knows, because as you know, many HSP patients have not been diagnosed yet. Every gene has been sequenced, that's known, but we still cannot find the underlying cause. So there have been estimates that it may be 200 HSP genes in the end. Today we have about 100. And often they get a number, and you may know the number that your HSP gene has. But there are also now many HSP genes that don't have a number, because frankly, we found that giving a special number may not be all too useful when we already have a gene name. So it can be quite confusing. So despite all this knowledge, in the sort of best laboratories in the world, when we sequence HSP patients, we typically say that in about a third of these patients, we cannot find the underlying gene, okay? So clearly there is more work to do. We want to get at least to say 90-95% of diagnostic rate for patients. But this is sort of in the academic situation where we can do things that in the real world maybe cannot be done, or where really expert physicians look at patients and make sure they have HSP and not something else. Now, just very recently, a study came out from one of the largest clinical testing labs in the United States. This is a company, and oops, it's called Invitae, and you may know it. And they looked back at 25,000 patients they tested over the last few years with large gene panels. And I call this real world results, because all kinds of neurologists and physicians would send samples to them that they think should be tested for HSP. And their rate of diagnosis is actually less than 15%, which is astounding to me. There may be a number of reasons why it is that low, but it can certainly not fully explain the large difference between 2/3 and say 15%. So in real world, there may be even more HSP patients out there that they don't get a genetic diagnosis today. And some clarification here. So gene panels, this is what you usually would get from your doctor prescribed as a first step of diagnosing which type you have. And these panels really contain the HSP genes that we know, so about 100. Sometimes they contain genes from related disorders, but it is a relatively small number because, as I said, there's some 20,000 genes out there. So when this comes back empty-handed, sometimes insurance companies would pay for what we call an exome sequencing. So that is basically a technique that can sequence all 20,000 genes in the genome. So theoretically, an analysis of an exome should give you an answer, right? But keep in mind, even that very large amount of data represents only 1 to 2% of the entire genome, okay? So that is also quite astounding, because the next step up really, if you really want to know what is underlying your disease, you would have to do something we call genome sequencing. Now, genome sequencing, truly, the idea is to sequence the entire genome of a person. That would mean all the 20,000 genes, but also the space in between. But even genome sequencing, as it is performed today, can only capture roughly 92% of the entire sequence. So there are still regions in the genome that are very difficult, for technical reasons, very difficult to sequence. And also this tells you this big difference here between exome and genome sequencing, that the space in between the genes is much, much bigger than the genes themselves. And frankly, we don't really understand very well in genetics yet this space in between the genes. We don't quite know what it means. But arguably, a lot of the missing HSP genes, patients that don't get a diagnosis, it is because something outside of these 20,000 genes could cause it. So this is really where research is now heading to create a lot of genome sequences from HSP patients and trying to get much better at understanding what else is there outside of these genes. So why is it so important to know your HSP gene? Well, here are some answers that come from patients or could come from patients. First of all, it definitely gives you a diagnosis, and it gives you a diagnosis that is very specific, and it is really the basis of everything that all the considerations that may come after. So for instance, once you know your gene, for instance, SPG4, you can go out on the internet or with the SPF or with your physician and find very detailed information about this type of HSP, because the different types, they play out somewhat differently. And knowing your type can give you good information on what is to be expected in the future and so on. Very important to patients and families. When you know your gene, you can seek advice, professional advice, and they can tell you what actually is the risk to pass a gene on in your family to your children. Also, if family members wish, it is now really easy and also affordable to do a diagnostic test just for that gene. And this could even be done if somebody wishes before the symptoms appear. And maybe most importantly, as you just heard in the other talk, we expect now in the field that genetic therapies that really target individual genes and try to fix them, will be the best solution for HSP patients. So the crux is with this, you have to know with high certainty what is your gene. If you do not know your gene, you will not benefit from these genetic therapies. And of course, when you know your gene, you may choose to really support research that specifically improves the research on your gene, right? So, and I want to convince you of this paradigm that there are really sort of two sides to it, and they're very interconnected. One is knowing the exact genetic diagnosis, not just knowing I have HSP, knowing the exact genetic defect is very important in itself, but it also enables us to develop and apply genetic therapies, okay? So it's a great opportunity to know your underlying cause, because then in the future, we hope, you can really benefit from genetic therapies. And of course, the opposite is true. If you don't know the genetic cause, you may not be able to use genetic therapy. And if somebody has a cause that's not even known yet, where we don't know the gene, maybe a novel gene that is still hiding, there will not even be a development for genetic therapy. So there are many genes, as I said, as you know, and unfortunately, it's also true, this causes somewhat fragmentation of the research, right? If you study, if you can focus on one target, that sort of focuses the entire effort. But now we have some 100 genes, and certainly there is some fragmenting forces here. It is still true, but it is definitely getting better that we tend to work in silos. We work at different universities, at different companies, on different continents. So that all drives us to not always sort of exchange what we know, what we see, and sort of work in silos. And also that finally leads to the fact that research is not always coordinated. And sometimes it's good that it's not coordinated because solutions may come from unexpected places. But certainly, especially for rare diseases, arguably it would be good to have some sort of coordination happening. So one answer we found in my group and collaborating scientists is to build a database, essentially, for genetic data from HSP patients. And we call this the Genesis database. It serves not only HSP patients, but also other rare disease patients. But it has been quite successful, and we're very proud it's been supported by some major foundations like the MDA. Just last week actually renewed support for this database for another 3 years. We also get support from other disease groups like peripheral neuropathy or what's called CMT groups as well. And when we look in this database and just look at the HSP data that is there today, and I really copied and pasted this today, this morning. First of all, you see on a world map that we have quite a reach into South America. These are real patients, real scientists who work with us on HSP. And actually, these scientists may have collected HSP patients, as we know, for instance, from North Africa. So that map doesn't even cover the reach we have, but it gives you an idea. It is very international. And you can see here that we have about 1,000 of these exome sequencing data sets, now about 50 whole genome data sets. And the majority of these data sets, about 90% really, represents patients that have not received a diagnosis yet. So using this platform, we can coordinate with many scientists. We look at the same data, we exchange ideas, we can alert each other to ideas and novel genes, but also outside, we collaborate with other groups that are there with similar efforts to do something what we call genetic matchmaking. And this has really, in the last few years, resulted in a lot of new discoveries. And here's one example, a very typical example where you see a novel gene has been published about a year ago called UBAP1. And this world map tells you which investigators, which scientists were involved and where they live and work. And it's remarkable to see that, of course, we have investigators in the United States, and in Miami we have the Genesis platform, which allows all this connectivity between investigators. But then we have Canada involved, we have Spain involved, and Germany, but also Bulgaria and Belgium, and also in Asia, we have Central Asia involved. So all this data comes together in this database and makes it possible for scientists to find that hidden signal. And that was published, as I said, and now a number of groups have confirmed these findings. So this is actually looks to be a quite important novel gene for HSP. Some detail here. Everything started really with a large family from Central Asia, from Iran, that has been studied over decades really, and you see multiple generations were affected, and that has really opened for us the finding of this novel gene, so that we now can even find it in individual patients who don't have any family history, like here and here and here, from Germany, from Spain, but also in some other larger families. What is sort of unique about this gene for us as scientists, fascinating, is that often mutations in HSP genes, they spread across the entire gene, and it's sort of sometimes difficult to decide which variants in a gene are really mutations or which are just there by chance. Now, this gene is a bit special because all the variants we identified, they all sort of they are what we call truncating. So they disrupt the gene so that the protein is not fully expressed. The full length of the protein is not expressed. And pretty much all the variants fall in a sort of very circumscribed area in the protein. And what this causes is instead of the full length protein being there, only this first bit of the protein will be in cells. And up here you see the protein structure, and you see these colored boxes. One says UMA. You see that the bit that's what we call UMA is especially important, is always preserved in these patients. So that doesn't get disrupted. But what gets disrupted is this other piece, which is also very important, which we call UBA. So these two UBA boxes, they always get lost in these patients. And that really gave sort of away the mechanism here for this disease, this type of HSP. And I think we have a pretty good idea what the problem is. This gene is involved in something we call the ubiquitin pathway. And essentially, what ubiquitin does, it is a signal that attaches to proteins that need to be removed. Like in the real world, in the cell, there are also always proteins or fragments that are sort of garbage that need to be removed safely. And that is facilitated through ubiquitin. It's basically a sort of garbage removal pathway. And that pathway is disrupted here. Or some parts of this pathway at least. So that effectively, what probably happens here is that the cell really cannot remove safely things that shouldn't be there anymore. And so over time they accumulate, and then they stress motor neurons, and that probably leads to those neurons, those cells, to eventually die. So moving on, as you're all aware of, genetics and HSP has quite a history now, over 20 years. And when you look at the genes that are known, that are being tested for, or when you ask your peers, your SPF, you will find that many patients actually have a mutation in the SPG4, SPAST gene, okay? So the SPAST gene accounts for a large number of HSP patients. About 40% is the estimate. But 60% or so of patients don't have a SPAST mutation. So these other patients, they have mutations in other genes. Other more common HSP genes are, for instance, SPG3A or REAP1, okay? So when you add up SPAST, SPG3A, REAP1, you may find that 60% now of all patients can be explained by them, but still there are 40% or so missing. And as you go on and add up the other known HSP genes, you find, generally speaking, that they get more rare, more rare, more rare. And for the rarest of HSP genes, really, we only know a handful of families, and sometimes even less than that. So that is the challenge we have. Genes get more rare, so they get harder for us to recognize. And the other side of that is we get sometimes a bit concerned that we, as everything sort of goes down and down and down here, when we add this all up, we may never reach, 90, 95, 100% of diagnostic rate. So that's sort of puzzling to us, and we don't have a full answer to that. But there has been very recently from our group a discovery from a related disorder. It's not HSP, but it's very related to it. It's called peripheral neuropathy, or sometimes CMT, or Charcot-Marie-Tooth disease. Okay, these patients, in these patients also motor neurons die, but it's not the cortical motor neuron you heard about this morning. It is the spinal motor neuron. So we have two types of motor neurons. So in these patients, the spinal motor neuron dies. Okay, and so understanding those motor neurons may be very important also to understanding what's going on in HSP. Now, this is a great example because, first of all, it was published in a very important journal. So professionally, we're very proud, of course. It was published in Nature Genetics, and Nature Genetics even decided to put this story on the cover. So what you see here, this sort of pink DNA structure is actually refers to this discovery. And I'm gonna explain you in a few slides why this is so important and why this may invoke some hope for where HSP may be headed to. So first of all, very similar in CMT to HSP. These are all the genes, the CMT genes discovered over the last 10 years or so. And very similar, these numbers, what do the numbers mean? They mean these were the number of families that were described in the very first paper. So whoever discovered a particular gene, these were the numbers of families that they prescribed in their papers. And it gives you some idea how rare or how common a gene may be. And as you can see easily, most numbers are very small. So as I said before, most new disease genes are very rare. Some are more common, but most are rare. Now here, the red arrow is our new discovery. And you can see, without looking at the number, this is really different. We were able to find something that affects quite a few of these patients. And this is exciting for a number of reasons, of course. So first of all, here you see how we discovered this. The idea was to look for potentially pathogenic DNA variation that comes up in multiple families with CMT. Okay, so we looked for this sort of signature. And here you see a sort of a snapshot. This is exactly what scientists would look at when they use this Genesis database. And we had a very talented professor here who discovered this in Miami from London. And he saw, surprisingly, that a lot of these patients with this disorder, they had one particular variant in this gene called SORD. Okay, so at the time there were no red dots or red flags that could make us aware that this is important, but clearly there were a lot of these sort of aggregated or enriched. So we followed up on this, and this led to the discovery of the SORD gene. And here are some families, and I show you this. First of all, you don't see any big family here, like I showed you in the other gene, for the UBAP1 gene. So that's remarkable for two reasons. Number one is often we don't have large families. Actually, mostly we don't have large families. So we are actually able to find these genes even with small families like this or individual patients. That's important that we're able to do that. And also interesting is that when you look closely, most of these families, it's really not a real, quote, family. It's just one person in that family who has it. Parents are not affected. Mostly they don't have siblings that are affected. They are the only person in that family that has it. And those are, for a number of reasons, the most complicated for us to diagnose. So we're very glad that we can find a lot of these patients with this new gene. And we were, of course, also very happy to see that when we then screened cohorts of CMT patients that about 10% of all the CMT patients actually carry this mutation, about 10%. So with one gene now, in 2020, we're able to explain another 10% of patients that have been looking for a diagnosis for a long time and nobody could tell them what they have. So this is a big advance. And what is even more sort of exciting is that we may have, or we have, a pathway to treatment. We're quite certain about this. So the gene SORT is really, when you spell it out, it's called sorbitol dehydrogenase. Okay, so, and we see these mutations in our patients. Most of these mutations destroy the gene. They destroy the gene, and so no protein can be there. So this is gone. What is very fortunate also, we know already a lot about sorbitol dehydrogenase. We know what it does. It's an enzyme. It converts sorbitol, which is a sort of a sugar, into another sugar, which is called fructose. That's what it does. And it does this because it is one way of processing glucose. So glucose gets turned into sorbitol by this enzyme called aldose reductase, and then sorbitol gets immediately removed by turning it into fructose. So when we take this away, this doesn't work anymore. It stops here. What happens? Glucose will still get turned into sorbitol. And now we have increased levels of sorbitol. And this is already known from other studies. A lot of sorbitol is not good for your nerves. And that is with great certainty. This is why these patients, why their motor neurons die. They have too much sorbitol in their nerves and in their motor neurons. So an idea how to fix this is actually to block the aldose reductase. If we would block this one in these patients, then glucose wouldn't be turned into sorbitol in the first place. Okay, so we would avoid a high sorbitol. And very fortunately, the pharma industry has worked for over 20 years in developing drugs that block exactly the aldose reductase. They're called ARIs. So there are a number of ARIs out there that can be tried. And in fact, this is what we do. We are working now with a company in Boston. They have a very modern and new ARI already in clinical trials. And this company is very excited. Within a few weeks, we came to an agreement with the University of Miami to test this further and also start clinical trials, probably even next year, which would mean we might go from a discovery of 10% of CMT patient genetics to a real treatment in less than 2 years. So, and just to say, this is not HSP, but it is very related to it. And we were extremely surprised when we found it. There are very fascinating scientific reasons why it was not discovered for such a long time. And the same reasons might apply for HSP. So I would not be entirely surprised if some more common HSP genes will pop up in the near future. And some of them may have a very fortunate situation like this one. And also, you may think, well, I don't have CMT, maybe I don't care so much. As you heard also in the previous talks, HSP, or we call it spastic quadriplegia, doesn't live in a silo. We used to think in the last 100 years that all these diseases, they have their own little island. In reality, they are connected. And some of you may have symptoms from something called ataxia. That's not uncommon for HSP patients. Some of you may have peripheral nerve damage, which is an overlap with CMT. And it goes on. And here you see ALS. you heard this morning how HSP is related to ALS possibly. And so understanding this and seeing the big picture is very important. And it also will lead, as you heard before, probably will lead to treatment of entire pathways, not single genes, but pathways. So you may be able to, with one drug, treat several different genes and several different diseases. So one way scientists try to study this better is to understand these pathways and make predictions about them. And we have also tried this and continue to work on this using sort of mathematics and network biology, and really what it is, powerful computers and very smart people to build models, genetic models for HSP and other disorders. And here you see such a model that connects these different genes and possibly, we hope, can pull out novel genes or can define pathways but much better to us. And we're also very hopeful that things like machine learning or artificial intelligence approaches may help with this. The fact of the matter is our brain is not made to remember 20,000 genes out there, but computers could easily process 20,000 genes. So we're trying to find ways to take advantage of this. And one outcome of this study, for instance, is that it is a very, what we call, unbiased approach. We take every gene out there in consideration. We take the entire protein connection catalog that's out there into consideration. And then we let algorithms and a computer tell us what is there. And what we find then is, surprisingly, what we already knew, of course, is that HSP and CMT, they overlap. They're not separate, they overlap. They're very connected disorders. Now you may say, that is maybe a sort of a lucky situation that you found this, but is it really true? Now, we looked at other diseases with the exact same algorithms, like cancer. There is no overlap here between cancer and HSP. Deafness genes, there is no overlap. Muscular dystrophy genes, there is no overlap. Parkinson's disease, no overlap. ALS, we see some overlap with ALS, not as strong as we may have thought we would, but ALS is definitely not completely separate from HSP. And also very much overlapping is something called ataxia. And I'm sure some of you know this already because they experience symptoms from ataxia. So to sort of, what this leads us to is thinking about therapy. And I mean, we heard this morning already very good arguments where therapy research is heading and how this probably is going to play out. But as always, it's difficult to make predictions about the future. But I think most colleagues now in 2020 agree that gene therapy may be the best way forward, because gene therapy can really target individual genes. It can replace genes. There are things like CRISPR. You may have heard of CRISPR, which in the future, we hope it can actually really fix genes, not just replacing them or suppressing them, but really fixing the mutation that is there. And in really the last 2 years only, the first gene therapies have come to the market in the United States and really in the world. And there are some absolutely stunning results in the spinal muscular atrophy field, which, by the way, is also a motor neuron disease. It's the spinal motor neuron, the lower motor neuron. But in principle, that will very likely work for HSP as well. So gene therapy is probably the most exciting topic that's there, but it's not the only topic. And so, you know, in the right situation, there may be things like enzyme replacement possible. sometimes we understand the mechanism, what goes wrong in a cell, and we can try to rebalance the cell metabolism. There is a lot of research going on cell-based therapies. The idea is really, when your neurons die, maybe there are ways to replace these neurons with stem cells or techniques that use stem cells, and somehow we can replace them. And where we are really today, but will certainly also develop further, is many of you, you experience certain symptoms, weakness, spasticity, and so on, and pain, that we have sort of a cocktail available to treat the symptoms in each patient. But that would not be a cure. That would not be a cure. We think gene therapy probably leads to a cure. And to be very clear with the gene therapy, this is a type of gene therapy that is only going to affect your personal cortical neurons. This would not affect the children that you may still get. This kind of gene therapy would not change the genomes of your children and their children. It is really something that would only play out in your own body. So from my sort of point of view as a geneticist, we of course want to continue this fabulous pace we have in discovering, understanding better the genetic basis of HSP. So to keep the pace up, even when genes get harder to find and they're more rare, data aggregation is going to be so important. Data aggregation, convincing our colleagues from different countries, from different research groups to share the data, and something like the Genesis database has turned out to be a fantastic force in finding these genes. So working together, of course, leads to data aggregation. And I already mentioned Genesis. And not at least, none of this would ever happen with the active help and participation from you, the HSP community. So getting involved with research projects is important, because only by studying you, the patients, we will really be able to understand what this disease is about. So to finish up, we live in most curious times with COVID-19. This is a recent lab meeting we had in my group, and we took this screenshot, and we continue working. As you see, people are in the lab, but a lot of people also work from home. Because we work a lot with computers and data, we can quite effectively continue working from home, so that is good. And as you know, I'm at the University of Miami. It's a quite stressful moment in our community right now with COVID, but we will definitely pull through and recover from this. And with this, I would finish my talk, and I believe questions will be answered later, and maybe I do this in person or by email. Just reach out to me. Thank you.